go on to talk about these more specialized controls. And I'll start off by talking about zymogens, also known as proenzymes. And these are defined as the inactive precursors. These are the forms that are synthesized. So they're synthesized as inactive enzymes or other proteins, because other proteins use this type of activation as well, even if they're not enzymes. They acquire full activity by the specific cleavage of one or more peptide bonds of the protein. Now, this seems to be a very odd thing to do. You synthesize an enzyme in an inactive form, where you have an intact polypeptide chain, and then you activate it by partially breaking it down, by cutting one or two or a few of the peptide bonds of the several hundred that may make up that particular enzyme. Now that's an odd thing to do, partially break down a polypeptide chain in order to do something good to activate the enzyme. But as we'll be seeing, what it does do is really uncover the active site and give you the active enzyme. Well, let's consider examples to see what this is all about. And I'll talk about the examples of insulin, the proteolytic enzymes of the digestive tract, and blood clotting factors. So insulin is generated by the excision of a specific peptide from what is called proinsulin, the original inactive form of insulin. The proteolytic enzymes of the digestive tract are synthesized as zymogens, inactive forms, in the pancreas and stomach, and then must be activated. And blood clotting factors, these are produced by the activation of zymogens, of clotting factors, that is, inactive forms of the clotting factors. So here is insulin. Now, insulin is not an enzyme, but it actually follows a zymogen type of activation. So insulin is activated by the removal of a specific peptide from the inactive proinsulin molecule. So originally, we have the synthesis from our genes of proinsulin. And this is a relatively small polypeptide chain. We start here at the amino terminal end with amino acid number 1. We go down and around. We go past 65, add 21 more, 86. Total of 86 amino acids in this chain. Anything less than 100 is very small. So this is quite small. And we have 3 specific disulfide bonds involving specific pairs of cysteine side chains, the SH groups of specific cysteine residues along the chain. We then have an enzyme come in that actually specifically cuts just 2 of the peptide bonds of that 86 amino acid chain, between residues 30 and 31, and between 65 and what was 66. So we just cut 2 of those, and then we remove that middle third or more than third of the original protein chain. All those blue amino acids are just thrown away. And what you have left is the 2 other chains that the light brown and the reddish colors here. So this is actually the active insulin. This is the active insulin here, consisting of 2 very short chains. One of 30 amino acids, the B chain, one of 21 amino acids, the smaller A chain, A and the B chains. But these are held together by 2 of those 3 disulfide bonds. Because originally, those 3 disulfide bonds were all intra-chain. But now, after we remove that middle portion, we have 2 of those disulfide bonds are interchain, because they're between the 2 small fragments of 21 and 30 amino acids. So those 2 disulfides help to keep those 2 chains together. insulin. Another thing that should be kept in mind is that the active insulin does not have two linear chains as we see here. They're just drawn as linear chains for simplicity. Like any protein, even though these chains are very small, it doesn't behave like a protein. Like any protein, you get a three-dimensional folding of those two fragments. You don't get a lot of structure, you don't get a lot of three-dimensional structure, you don't get a lot of alpha helix or a lot of beta strand, but you do have a little structure. And so the insulin chains are folded into a structure like a little protein. And those chains are kept together, so they are one little globular structure, not like two globular structures held together. It's like one globular structure that is active in its function. Now to talk about proteolytic enzymes of the digestive tract. Four of them in particular that consist of the active proteases called trypsin, chymotrypsin, carboxypeptidase, elastase, and pepsin. But all these active forms are, first of all, synthesized as inactive proteins, inactive enzymes. In other words, the zymogen forms, the inactive forms. And they go by names of trypsinogen, chymotrypsinogen, pro-carboxypeptidase, pro-elastase, and pepsinogen. So we have these inactive forms. The first four synthesized in the pancreas, and the last one in the stomach. And then, during the process by which they will be secreted into the digestive tract, they're converted from the inactive to the active forms. The reason for this is self-protection, basically. We don't want to have these enzymes, which are secreted and synthesized in very large amounts, we don't want them damaging our own cells of the body. We want these enzymes just to be breaking down, just to be attacking dietary proteins, breaking the peptide bonds of dietary proteins, as all of these do. So these are all proteases that attack dietary proteins, but we don't want them to attack our own proteins. So we only want them to be activated when they're in the digestive tract. So how do we get this zymogen type of activation? Well, we get it as shown here. I'll just show one example, that of chymotrypsinogen being converted into the active form that is called alpha chymotrypsin. And it goes through a series of steps. We start off with one protein chain of 245 amino acids. We then have trypsin, so that's another one of the proteolytic enzymes, making one break in the protein chain between amino acids 15 and 16, producing what is called pi chymotrypsin. Pi chymotrypsin is active, but it doesn't attack dietary proteins. What it does is attack other pi chymotrypsin molecules by cutting another peptide bond and releasing this dipeptide consisting of residues 14 and 15. This gives us delta chymotrypsin, also active, but not active in breaking down dietary proteins, active in breaking down other delta chymotrypsin molecules by cutting out a second dipeptide consisting of amino acids 147 and 148. What we have left is then three fragments that we call alpha chymotrypsin. So alpha chymotrypsin, this is the active form. This is the form that will attack dietary proteins. And it's important to remember that disulfide linkages will be holding these three fragments together, and they will be folded into a three-dimensional globular structure that in fact looks a lot like the original three-dimensional structure of chymotrypsinogen. You cut out four amino acids, producing three fragments, but they stay together, uncovering the active site. That's the essential point. By cutting the protein chain in two locations, we've uncovered the active site so that it is ready to now attack dietary proteins breaking down peptide bonds of dietary proteins. So I'll leave that there for today. Continue on.